Hi, everyone. We have as our guest today Anna Ipata. Anna is stepping in for Mickey Goldberg, who was called into the neurology wards today. So I'm happy to have Anna with us. Anna received her doctoral degree, MD, from School of Medicine at University of Pisa. During her medical career, she focused on the diagnosis of cerebral palsy in children. And after several years of medical practice, she transitioned to research and obtained her PhD in neuroscience from University of Verona, where she studied neurophysiology and non-human primates. Then she came to the United States, joined Mickey's lab at NIH, and came to Columbia University when Mickey moved his lab from NIH to New York. Since then, she's been leading the work on understanding the relationship between learning of sensory motor associations and recording from the lateral cerebellum as well as the dentate nucleus, and analyzing the relationship between these regions and the cerebral cortex. Anna, thank you for being with us today.
Thank you so much for this invite. Okay, so why the cerebellum? Why? So this is, of course, the topic is the cerebellum behind motor control. Of course, we know that it has been recognized for its involvement in motor control and motor learning through its connection with the motor arm. But we also know that cerebellum reciprocally connected with the prefrontal cortex. And we know that the visual motor association tasks are widely used to study the neuromechanism of goal-directed behavior in the prefrontal cortex. So there are evidence that the cerebellum is involved in the visual motor association task behind the eye blink conditioning. And for instance, in our lab, with the inactivation of the CRUS1 and CRUS2 that are in the posterolateral cerebellum, we know that it impairs the learning of new visual motor association, but slow a previously learned association, but not the performance. We also physiological studies in our lab, we found that during visual motor association, the simple spike signal a cognitive error of the previous trial. So taking together this previous data, now we focused more in the sensory motor transformation. So what's happening from when the stimulus occurs, started, and the decision of the monkey. So why and what are the hypotheses? So the function of this. So we know that in motor domain, the cerebellum, at least this is the main view, broadly speaking, transmits anticipatory sensory feedback, that feedback predictions that are associated with a given motor command to the neocortex to facilitate a quicker onset of the movement. But is it possible that similar mechanism occurs during goal-directed behavior, so that it can transmit an anticipatory signal predicting not the motor response, not the output, the feedback of the motor response, but for a goal-directed behavior. So we recorded from the posterior lateral cerebellum during a visual motor association task. Very simple. So the monkey grabs two bars, and then there is a fixation point. And then one or two stimuli appears. One stimulus requires the release of the right bar, and another stimulus of the left bar. So we trained the monkey to perform this task called the overtrain task. So the monkey, every time we present this task, he does this 100% correct. So this is an overtrain task. After recording about 30 trials, we switch to new symbols. So the monkey has never seen before. Now, one symbol, the motor response is always the same, but the sensory motor transformation has to change because the monkey has to learn by trial and error which symbol is associated with the right and which with the left hand. Again, the movement is the same. So this is a session of one recording. This is the behavior. So we use in the x-axis is the ordinal number of trials, And then you see the dots, the black dots at the bottom is when the monkey make a wrong decision, and at the top, a correct decision. From from the new task. So you can see that this is quite to the monkey makes a lot of errors, but then drastically, okay, he started to learn, so he does only correct. So instead of using beans to analyze the behavior, so the learning curve, we use a logistic regression. So we can pinpoint that time when there is this switch. So looking at so this is the logistic regression for this binary logistic regression of the performance. And we divided according to the threshold. So in early learning and late learning. So with early learning, we used the trials that are below when the logistic regression the threshold is 0.6. And then the late learning when the threshold is about 0.75%. So when you're talking now about early and late learning, this is what I refer to. Okay, so we recorded from the posterior lateral cerebellum, cruise 1 and cruise 2 in 2 rhesus monkeys. And we recorded the Purkinje cell, classical simple spike, complex spikes. So we knew that we are in the cerebellum. This is a typical activity. Okay, and we recorded, and then, so now, again, this is an example of one neuron in an overtrained task. In the left, when aligned with the stimulus, in the right, aligned with the onset of the movement, and this is the spike, the firing rate, and these are divided when correct trial, when the monkey release the left or the right hand. So for now, we are not talking about left or right, but only to show that we are focused now in the time during the visuomotor transformation, while in the previous paper or error, we were focused on what's happening after the movement. This is before the movement. Okay, so these are 3 neurons recorded during the overtrained task. With the left hand response correct, that is the blue, and the right hand response correct. You can see that these 3 neurons have a completely different pattern of response. So this neuron responds both to the left or to the right, it doesn't matter for now, but at different time windows. So we cannot have we cannot do a bigger population because the timing is different. So, but anyway, so this is overtrained task. So the question is, okay, what is the signal? So these are now the same, no, so we can say so individual CSD cell encode both left and right hand association. Now is independent if one is higher than the other, but in a wide range of temporal dynamic. So what is the signal? Is a motor signal, a sensory signal? What is it? So to answer this question, we can see that this is the same neurons in the early learning. So this neuron on the top during early learning has a completely different profile of response. There is no difference between left and right. There is not this peak before the movement. The neuron in the middle is not quite different. And the middle at the bottom, it's completely noisy. Okay, so already this analysis shows that, okay, so maybe it's not a pure motor, because remember the hands response, motor response is always the same. Now, in late learning, we see that there is, again, a temporal change, a change of the profile. For instance, the number 1 neuron, there is a quite increase of the activity before the movement, even if in late learning doesn't go back to the same overtrain. The second neuron that was not selective, for instance, for the hand, neither in overtrain or early, now acquire a selectivity that is in which you can see there is a inhibition when the monkey releases the right hand. And the bottom, it seems that to almost approach the overtrain, the overtrain task. Now, a point that I would like to stress now is this. Okay, maybe the difference between overtrain and late or early learning is because, yeah, this stimulus is different. So this is, but the fact that we found, remember, from early learning and late learning, the symbol is the same. So if there is a difference, it means that it's not a symbol and it's not a movement per se. Okay, so we can say that Purkinje cell maintained a response to both left and right hand, but the activity pattern shifts from the overtrained to the not. So the response of Purkinje cell in the overtrained task may reflect the acquisition of the correct sensory motor association and non-motor command. Okay, so how do we analyze? Okay, so because of this heterogeneity of the temporal dynamic, we cannot do a population analysis, put all the neuron together. So what we did is to see the response of the left or the right hand compared to the baseline. This is an overtrained task. Each line is one neuron, and is shaded of blue and red, and these are the p-values of the t-test and multiple comparison, all the, I don't want the method, but anyway. So you can see that the significant activity between the onset with the response after the stimulus, before the movement, and the baseline change. So the left is, for which we have aligned with the stimulus, aligned with the movement. S is stimulus, M is movement. So the onset, the latency when the neurons discriminate between one trial and the baseline change in a temporal, but I align here with the peak of the maximum difference. So what is important to see, so across the population, we have that the left and right association are identified at different times, but for each neuron, but collectively, almost from the onset of the stimulus to the end, there is some information. Now, what's happened, sorry, lateral learning, look at learning, I just missed up. When a monkey is learning, look at early learning, so there is still a response of left or right, but much less neuron has a response. While the lateral learning in the middle, so the neuron reacquire kind of a response to the left and right. What I would like to say is that the neuron in the left and the right and are aligned with the onset, but doesn't mean that, for instance, the neuron number 5 in overtrain is the neuron number 5 in late learning or early learning, because I can see there is also a kind of a rearrangement. So neurons are not selected to become selective and so on. So we can say that individual Purkinje cell responds to both ends, but a temporal window, a different temporal window. And the number of neurons engaged increase with the performance. Now, what we did here, so we take the timing for each neuron when the neuron identified the left and the right compared to the baseline. And this is an empirical cumulative distribution of this time and across all the neurons. So OTE, you see, is the black, for the right and left hand. For the left hand and the right hand, you see that the identification of which hand through the response occurs in the early learning and in OT much earlier than in late learning. So late learning starts later. Now, this can be the reaction times, of course, but if we align with the movement, this shift is still present. So that means that this shift is not purely motor, is not related with the reaction times, but has something to do with how consolidated the stimulus response association order is consolidated. Now, but what we were very interested is that what happened during the child discrimination. So we see that like in this neuron, this is an overtrained task, left and right correct trials. There is a difference between the left and the right stimulus association, okay? So what we did is we analyzed with the sliding window temporal, the time when the difference between left and right association start and also the duration. So in the next plot, you can see this, all these P, okay? So each of a line is a neuron. This is an early learning. And then the shade of black are how significant are the P. So in the early learning, few neurons distinguish between the right and the left hand that we call choice. And then, and also very late. So from the onset of the stimulus, that is the left, and the movement. Now, in late learning, so we have, these are all correct trials. In late learning, more neurons are engaged and are recruited that differentiate between left and right hand choice. And also you can see also from here, the discrimination between left and right start earlier from the onset of the stimulus and the movement. And everything is quite before the movement. So, and in this is the overtrained. So when monkey knows, much more neurons are engaged and differentiate the choice at different time. So there is a recruitment also here of neurons which can discriminate and is a learning dependent way. So now we did exactly the same. So the empirical community distribution of the time when each neuron discriminates between the left and right. So with the encoded dynamic, because as we said before, so even if one single neuron identify the time at a certain point, but this is not enough, is the collective activity of all the neurons. So this is the empirical community distribution, of the, again, of the time when the neuron discriminate between the left and right choice in early learning. This is from the onset of the stimulus. We can see that's only from the onset of the stimulus, like 200 milliseconds, very few neurons can distinguish between the two choices. But in late learning and also in the overtrained task, from the onset of the stimulus at 200 milliseconds, already 60% of the neuron had identified. But again, this can be the reaction times. But if we look aligned with the movement, this shift is still present. So the early learning encode the choice, which is the left or the right association with the stimulus, very close to the movement. While in the late learning OT, much earlier than the movement. So it's not a movement reaction times related activity. So in early learning, choice discrimination occurs close to the movement onset. And as learning progresses, choice discrimination occurs well before the movement. So now, until now, we analyzed the correct trials, okay? So the hypothesis was, okay, if during learning, the child discrimination, okay, it's related with the choice, so with the how strong is the visuomotor association, that means that during early learning, the correct trial, the wrong trial are treated the same, because even the monkey makes correct trials, in any case, it doesn't know yet which will be the output. So we compared the correct trials in early learning, so the choice discrimination between the correct trial, and the wrong trial. And we found that approximately the same number of neurons are engaged. And then we did, again, the time when in early learning, the neurons identify the correct or wrong association is the same. So that means that in early learning, the Purkinje cell cannot distinguish between correct and wrong, but in late. So we know that's, okay, we are seeing that is the population that's encode the choice, called the stimulus association, not the single neuron. So the best way to analyze is do a population. And so Valeria Fascianelli of the Theoretical Center, she did the analysis of this neuron with a linear decoder. So a linear decoder then provides more detail about the population temporal dynamic. This is a graph from the onset of the stimulus. Each line is the accuracy of the linear decoder to distinguish between the two choices. In the overtrained task, that is the blue one, you can see that's like the same that we see also with the population in physiology. That's about 200 milliseconds after the onset of the stimulus. The decoder identify already almost as accuracy, almost 100%. But look at early learning. At the same times, 200, 400, in early learning, these are correct trials. There is not identification of the left and right. In late learning, he almost approached the overtraining task. But what is also interesting is that, okay, what about, again, is the reaction time? What about according with the movement? So this is aligned with the movement. You can see much better here that in early learning, the decoder overpass the threshold of 60, this is the threshold, the chance, very close to the movement, okay? And the peak, it's about close to the movement. But in overtrained task, the blue one, and in late learning, that it's the identification, the decoder is already crossed the threshold much earlier. So again, it can be the reaction times. Another thing is this. So it's through this task that we are using, the stimulus and the movement are linked together. So you don't know what is, how during this process, the Purkinje cell identify the stimulus and the movement, because it has to be identified both of them. So we, in some neuron, we also recorded, after the monkey learned a new task, we reversed the stimulus and the response. So the response is already the same, and now the stimulus done a new task that he learned is associated with the right hand, now is the left, and then is the right. So that means the early learning, so we can disentangle the response to the stimulus independent of the movement, and response of the movement independent of the stimulus. So Valeria, she did the same analysis of linear decoder, and she disentangled these two information. So on the left is the accuracy of the decoder to distinguish the stimulus only, and is the orange, or the response. So in early learning, you see a line with the stimulus. In early learning, you see that there is not much different after the answer of the stimulus between the decoding of the stimulus or the movement, okay? So the visual stimulus is decoded only when the choice is made. Of course, then aligned with the movement, that is the right, sorry, there is not a label. Of course, aligned with the movement, the stronger the identification of the movement. Okay, so this can be poor at the point, poor movement. What's happened in late learning? In late learning, you can see that after the onset of the stimulus, both the stimulus identification and the movement are decoded much earlier, so they cross the threshold much earlier. But what is interesting is that the decoding of the stimulus occurs earlier than the decoder of the movement, and also remain quite high until the movement. So as learning improves, the visual stimulus is decoded earlier than the choice. Okay, this is the first conclusion, the summary of this. So we found that in overtrained and late learning, the majority of neurons respond to both hands and distinguish between the two associations. The population identifies the correct choice well before the movement onset, and the different firing rate patterns that we observed may reflect that different population of Purkinje cells within the cerebellum may be engaged in parallel process of different aspects of the task, potentially contributing to the overall computation of the sensory motor transformation. During learning, so we have a fewer neurons response to both hand and differentiate between the two associations, and the population identified the upcoming choice much closer to the movement and does not distinguish between correct and wrong. So we think that the recruitment of these new neurons during the learning can be allow the Purkinje cells to adapt to a new formation and change the task demand, to a dynamic reconfiguration of the neural circuits. Now,
Anna, do you mind if you ask some questions?
absolutely, no, actually, yeah, stop me because
Yeah, so let's see if there are any questions from the audience. I guess I'll start first. I didn't quite understand, is the P cell activity after learning the new task comparable to that cell's activity during the overtrained task?
no, that's okay, that's the point. So let me see. If you this here, okay, the left, the first column are 3 neurons in overtrained task. But when you go to early learning, you see it's not comparable anymore. It's different.
Yeah.
And in late learning sometimes also. If you see the third neuron, you can see that it's not selective in overtrain, also in early learning. But at a certain point, you see that there is an inhibition when the association require the right hand. So this neural change also is not a return to the previous one. The third one, no. So the third neurons actually seems that's then realigned with overtrained. But the many neurons, I would say that, no, I want to say the majority of neuron, but really, they can change also the selectivity for one hand or the other. And then there is a complete reconfiguration. So we have that in early learning, even if the neuron responds to both hands, many of them, they do not distinguish between the left and right. In late learning, there is more neuron. The same neurons start to distinguish between the left and right, but it doesn't mean that they go back to the overtrained task. So there is it's not necessarily this. In fact, in the let me see the choice. So here, okay, each line is when each neuron, a neuron start to identify the left with the right hand. If you see the overtrain at the bottom, you can see that across the population, each neuron contributes differently at a different time during the task and start to discriminate between left and right association in overtrain. Now, if you look at early learning and late learning, okay, in early learning, neurons still respond to the left and right hand, but there is no difference, okay? And then in late learning approach. Now, again, if you take, I don't know, neuron number 50, okay, in overtrained task, start to discriminate, I don't know, about 300 milliseconds before the movement. Doesn't mean that it's the same neuron number 50 in late learning. And also because here I aligned with the peak of the difference, okay? This is the real align. If I don't align this way, it's a mess because it's not the same neuron. If I align the neuron in overtrain and then I take the same neurons and I plot late learning, it's not like overtrained anymore. It's
okay, thank you so much. Yeah, I understood
okay.
that.
Okay,
There are a couple more questions. So let's go to Rich. Go ahead.
Yeah. Actually, this slide's perfect. Just two detailed questions. One is what in overtrained, what's the average reaction time is the first in the.
It's definitely shorter. Let me see, we have the reaction times in the decoder. You see in the top is the mean reaction times, okay? So in overtrain, it's about 40 milliseconds 35, in late learning, it's 100, and then it's definitely there is a difference.
Okay, and then the second one was on those early latent overtrained. Are there, I'm guessing that there's a lot of difference in the number of trials that go into each of those analyses, that there aren't very many early trials relative to the late and the overtrained? Is that correct? Or
Oh,
do you use the same number of trials for each of those spins?
okay, yes, of course. But when we did the analysis, okay, this is the analysis I did. Okay, I take.
No, I mean on that
okay,
slide that you show with the
yes,
choice encoding, for instance.
it's there is a difference of number of trials, but when I did the analysis, I correct also for the number of trials. So I did, I shuffle with the same number of trials, and then if I have I don't know, 14 trials for one and 7 trials for the other, and I found that is significant, I do the convalidation, permutation, bootstrap, okay? So to wait for the difference of a number of trials, of course. I mean, this is, I didn't say the method, but this is the method that I did.
Okay, thank you.
I didn't take a P like, okay, that
Okay.
convalidate this way.
JR, you have your hand up. Please go ahead.
Hi, Anna. This is Jennifer Raymond. I thought maybe I'm misremembering, but I thought earlier in this project you had seen evidence that this Purkinje cells responded kind of while learning a new association, and then that response went away. So are those cells just kind of part of this population, or am I totally misremembering that there were neurons that selectively were active when the monkey was learning a new association?
No, these are exactly the same neurons. Exactly the same neurons. The difference is that previous, we focused on what's happening after the movement. So we did analyze from the stimulus to the movement in the current trials. So we analyzed from the movement of one trial to the next trials. So that means that actually, if we find an error in that analysis, we don't know. So we have a movement, but then the next trial, of course, we don't know if we stimulus, if it's the left and right, no, okay? We just see that there is an error after. And this error after the movement, okay, decrease with, the difference decrease with learning. Now, here, I was focused not what's happening after the movement, correct or wrong, but during the association. So here, if we reanalyze the same data, but this time we focused on what's going on when the monkey has to decide which hand, independent of the previous trials. This is we don't know. It's independent of the previous one. Okay.
Have you? It would be interesting to know, like, the ones that show that transient activity. What are they doing during this earlier time in the trial?
You mean earlier time in the trial? What I don't understand. Maybe
the neurons that have that sort of delay period post-trial activity when they're learning a new association. What are they doing at this earlier time during the stimulus and the movement?
Yes. So I will show you also later, but it's something like that. Actually, there are neurons that's even the Purkinje cell that's also before the movement so there is a trial before the movement. They have a difference between if the movement will be correct or wrong, but relatively low. And
is that what we see in this slide? The red trace is lower than the two blues?
Exactly.
Exactly. All right, great. Thank you.
Remember, these are only correct trials. I'm analyzing here correct trials only in early learning and late learning. So when monkey doesn't know, even if it does correct, so for instance, the red, it's when monkey responds correctly, okay, in early learning. So that means that's the reason we are focused during this period between the stimulus, the movement. The monkey responds correctly, but it's by chance. It doesn't know. So these are correct trials, all of this. So that means that in the blue, in the pale blue and in the red, it's exactly the same kind of trial, the same stimulus, the same response. So correct in early learning, correct in late learning. But the difference of the population, the discrimination between this is not a discrimination between correct and wrong. This is a discrimination between which goal, which action will be made. So it's exactly the same. And it's not, it does not depend on the reaction times. And the reason is that if you align with the movement, if it depends on the reaction times, at the movement or before the movement, all of these curves should match because it's purely motor, but they're not. So it's in early learning, the red, if you look at the red, look at when the red crossed the chance of the decoder. It crossed the chance about 200 milliseconds before the movement. So 200 milliseconds before the movement, let's say the neuron probably is a chance. So this is, I think this is a pure motor response. But in the late learning, the pale blue, okay, if you can see at 200 milliseconds before the movement, already 80% of the neurons had discriminated what, in my opinion, if is the next response will be correct or not. This is, I mean, my hypothesis. So it's like my hypothesis that this is like a prediction, okay, of the outcome of the next response that has been acquired during learning. Because it's much earlier than the response. The neurons identify the correct choice. So my hypothesis is that, but then of course, super open, is that during learning, monkeys guessing, and there is the associations, the transformation, stimulus response transformation is not yet done. So it's just stimulus and response, okay? And then because the monkey, before also a correct trial, cannot predict, doesn't know if it will be correct or not. But in early learning, late learning, and in overtrain, yes.
Okay.
I don't know if, okay.
So Anna, there's one more question. Yoshiko
Yes,
has.
Yeah, thank you. So I think I'm might just asking same thing, but if I see the reaction time and peak velocity and all like motor aspect will be different in early learning and late learning, right? And so if you compare like same latency of early trial, early learning and late learning, are you going to see difference? Or like if you compare short latency trial and long latency trial in the early learning period?
Yeah.
Like, yeah.
Okay. Of course, this was an analysis. I tell you immediately, it's very difficult to find a balance of same reaction times because, I mean, in late learning, I cannot find it's very difficult to have a short reaction times. So it means that the analysis can be unbalanced. But for the number of trials, it's just it's only a matter of a number of trials. So if not, cannot give a strong statistic. But the fact that if you align with the movement, okay, if the difference is purely the reaction times, you are at the movement, so is the movement. So before the movement, it should be the same. Okay, because in any case, you have okay, the reaction that, okay, if the neuron at a certain point discriminate and responds, okay? So if it's long reaction times, suppose that the discrimination is all at the same time, on reaction times, the discriminate at 200 milliseconds before the movement, okay? If a short reaction times, the same, 200 milliseconds. So if you align with the reaction times, you should find that that's between the reaction times and the time when the neuron is fixed. Is fixed. So this is the same things that we do when we want to see if there is a difference, in the motor response. So when you align with the motor reaction times, if it's related with the motor reaction times, you should find a similar answer. Because if the reaction time dictates when the movement, so when the movement will be, when the decision, so it should be independent of the reaction times.
Okay.
Because
Thank you.
the gap between, okay. So that's the reason we think that it's not the reaction times.
Yeah,
Because-
thank you very much.
Okay, if there are no other questions, Anna, do you want to continue?
For me, yes. Okay. And then here we see that, as I'll show you, it's that there is, let me see, so discrimination of the stimulus before, when, during, okay, in late learning, sorry. So both stimulus and the movement are discriminated. And I think I went through here. So we had, yes, the recruitment, during learning, and then, yeah, we will have time for a discussion because this is not a long talk. Now, okay, we found that the Purkinje side that has been studied with long about for this, but it's only the learning or the visual associated cerebellum cortex. So the Purkinje cell are the only neurons together, of course, with the groundwater cell that's learn. And so we know that the Purkinje cell, actually the output is not the final output from the cerebellum. So this information has been converged. And where they converge to the deep cerebellar nucleus. So inhibitory impulse for multiple Purkinje cells. So for probably from Purkinje cells, they have a completely different kind of the response. They converge, you know, about 40, 60, converge at the single neuron. Not only this, so it's the deep cerebellum that then has to do the integration of everything. So, but not only this, we know that the cerebellar nuclei, this is in mice and then, and not, by the way, the cerebellar nuclei receive directed excitatory input from ossifiber inferior lateral, the same input that then it goes to the granular cell and then to the Purkinje cell. So it has the access to the same information that arrive to the cortex, okay, at the same time, so maybe also earlier. Not only this, but we know that has been found that some internal feedback, excitatory and inhibitory internal feedbacks from the cerebellar nuclei to the cerebellar cortex through the granular cell and to the inferior lateral. So, and then of course, they go back to the Purkinje cell. So there is also this loop. Okay, so the cerebellar nuclei. Then we also, another thing is that has been found that cerebellar nuclei, that they are found in, I think, fastigia, that is not really, there is not a fixed topography. So that's one zone, a functional zone, sent exclusively to a particular territory of the cell. But there are, I think, 50% in which the same neurons receive a convergence from different functional, different zone of the cerebellum. So there must be also an integration of signal, maybe functional different signal, because it's not so divided. So there is this convergence. So, and also we know that the cerebellar nuclei are involved in acquisition and conditional response and blink conditioning. Okay, so taken together, this means that, okay, so the next step, I thought, okay, we know Purkinje cell, let's see what's happening in the cerebellar nuclei, where we choose the dentate, of course, because that's the one that we know is connected with the frontal area. And the idea is that, I mean, not this, one of the idea of the people who study is that actually there is a function of cerebellar nuclei, and this, the function probably serve as a consolidation of the learning association, but also in learning, okay? So we recorded from the dentate nuclei, mainly from the dorsal, but I tell you now that until we do a real histology, it's not easy to say because it's not easy to say the dentate, but we are quite, so, but anyway, it's the dentate nuclei, probably the caudal part. So we did the same task, and then the overtrained task, and then we do the new task and the reversal. In particular, in this monkey, it's only one monkey. Since it's very, very fast with the new task, you learn it very, very quickly, so we don't have enough trials in early learning. So reversal, of course, it gets more new. So this is a 2 session of one recording. Again, the top is the new task and the reversal when we switch. It's again the correct ordinal number of trial to x-axis, correct at the top and wrong at the bottom. And even here, you can see that the monkey, after a certain number of trials, is switched immediately doing correct. So that is the reason we even here, we use a logistic regression. So to pinpoint the time when the monkey switched between correct and between the learner. So this is where we record from. This is our monkey. You can see the dentate. That it's and then we use brain sites, and so this is roughly the area of the dentate where we record from. Again, since not easy, it's I cannot tell you precisely in the dentate where we went because of this not easy way, at least for now. So this is I can say a typical really response to the dentate. This is a basal activity. So this is not actually a real response. This is so many neurons, and this is something I would like also to discuss with you about. Many neurons, they have a burst of activity, very lucid, by interval of 2 milliseconds, and this is probably because we know that the nuclei, cerebral nuclei, they have a higher spontaneous activity, and then sometimes a tonic activity. Okay. Anyway, this is a stimulus kind of a response. So very, very bursts of very, very super high activity. Okay, these are again 3 neurons recorded in an overtrained task in the dentate. Even here, you can see there is a very heterogeneity in the sorry. So this is the left and right again, blue, left and right, red, aligned with the stimulus. The stimulus here, it's many neurons, most of the neurons is 80 milliseconds. There are some neurons longer, but here, so I put longer. But in any case, when we analyze, we take into account this, of course. So here, there are 3 neurons. When the monkey responds, we respond sort of with the left hand or the right hand, correct trials. And even here, you can see the pattern of response is different. There is a typical burst before the movement in the first one, and then also an increase after correct trials. You can see the second one, also the third one, there is an increase of response when the monkey will release the right hand, but not the movement. And the third one, again, this is for the right hand, so this is definitely selected with the right hand, a peak and inhibition before the movement. So I didn't yet analyze the activity related with the pattern on activity that I think is very important here. By the way, I analyze now the firing rate. So the first question was, okay, we know that we see the Purkinje cell, there is this arrow signal after correct and wrong, then this tile. Okay, so the question was, this arrow signal is transferred from the Purkinje cell to the cerebellum nucleus. And so in this case, we should see that if there is an arrow signal, this arrow signal is later than the Purkinje cell. So here are 4 neurons. I divided the left and right, the response to the left in correct during early learning, correct and wrong trials. So correct is the solid blue and wrong the dark blue. So this neuron, first of all of them, you can see the profile activity. There is not difference. This is aligned with the movement, of course. There is not difference in the first neuron. There is a slight difference, but in the second, there is after the movement, in the third one, and in the fourth one, there is no difference. The same neurons for the right hand. The first neuron has clearly a decrease of activity in the wrong trials at about 200 milliseconds after the movement. The second one too, even if a different way, there is decreased activity. The third one doesn't have any difference. That is what we found there. I think it's by chance. And the fourth one, there is clearly a high activity during the wrong trials, only for the right hand. So The idea was because when I saw this neuron, I said, oh, maybe it's the higher, arousal, blah, blah, blah. But the fact that we don't find in the left one, it means that it cannot be. So it's related with it. So now, individual neurons in the dentate encode the outcome of the previous trial. Again, this is aligned with the movement, okay? So, and for one or both the right-hand association. So here, so I divided left and right, of course, because we cannot, so for the correct and wrong analysis. So at the top is the same analysis I showed before. So each line is a neuron. And then the shade of the blue is when the neuron discriminates with correct or wrong, okay? And then in the left hand and in the right hand for the, is early learning for the right hand. So even beside the fact that we have that's a neuron, some neuron has a different signal only for the right or the left, but across the population, you can see that this tied up whole arrow signal. And then we, of course, will discuss what's happened before the movement. But it's tied or the trial, before and after the trial. Of course, there are more. So now, the population encode the outcome, but of course also before with the previous for both left and right association, but in a dynamic way. Now we compare with the Purkinje cell. Okay, this is together. I put left and right together, I mean only left of the neurons, and then of the data neurons. So it's the same figure. So when the neurons discriminate between left and right, and is aligned with the sorry, not with the correct and wrong, aligned with the movement. The movement is when the red line is, I don't know, is shifted, but actually it's the red line, vertical. So this is and these are the same Purkinje cell that we analyzed for the previous papers, and the same thing. So correct and wrong, discrimination of correct and wrong. Okay, so the majority of neurons in the Purkinje cell discriminate this after the movement. Well, after the movement. Now, how is the timing? This was what I was interested. So which one goes first? So this is again the cumulative distribution of the proportional neuron that had distinguished between correct and wrong. In the dentate is the magenta, and in the Purkinje cell. So before the movements, very few neurons in both the dentate and the Purkinje cell. This is the question that's so discriminate between correct and wrong. After the movement, you can see after 100 milliseconds, around 100 milliseconds after the movement, more neurons in the dentate has already discriminate if the neurons, the response was correct or wrong. So it means that the population encoded the outcome of the previous trial in the dentate earlier than the Purkinje cell. Of course, these are the population, so it doesn't mean that all the neurons, they do the same. But across the population, some neuron, okay. Now, so this is the, now we compare also the choice discrimination with the Purkinje cell that we.
And if I can make a comment here. So
yes.
here you're looking at the simple spikes in the Purkinje cells, right?
Oh yeah. Sorry, yes, it's only simple spike. Yeah, and these are correct trials. Sorry, no, these are right versus and the wrong trial. Yes, this is only simple spike. Again, in the Purkinje cell, in the dentate, it's the firing rate, okay? And then I don't know if you want to talk about this, but there are these bursts of the firing rate in the dentate that really make the very strange profile, because you see really, even when you do the firing rate of spike density function. So I never analyzed the burst. I can tell you that I saw this burst more at the specific time during the trial, different from neuron, but still didn't make any analysis, specific analysis. From looking at the rasters, clearly that the bursts are not really random. So This is
Just
something I will.
confused. If you're thinking about the outcome of the trial in the Purkinje cells, why are you looking at just the simple spikes, not the complex spikes?
Because it's a different analysis. So first of all, because okay, if I want to compare the dentate with the Purkinje cell, I have to do the simple spike, because it's if I want to compare the complex spikes, then I have to find the dentate. So my point is this. So I don't know. First of all, in the dentate, I don't know when the complex spikes are received or from inferior lava. I don't know when, because I don't have I cannot discriminate between the fire rate. But I saw that in the literature that they found that actually indirectly, you can say, with the burst and pause. So pause, burst, they say can be complex spikes related activity. So indirectly, probably, if I analyze the burst, okay, or the probably it can be that indirectly is the complex spikes and or the from inferior lava. But so at that point, I can't compare, because if I want to compare the dentate with the Purkinje cell, it's with the complex spikes. Even because in that case, I have to record both at the same times. These are recorded at different times. So no,
Okay, thank you.
Okay. Okay, so the same we did for the choice. Okay, so the same analysis of Purkinje cell. So when the neurons start to distinguish between left and right. So again, these are the same neurons in overtrained task, different profile of firing rate, and then the peak of the response before inhibition. So it's very random. So even here, neurons that response to both left and right. This is likely Purkinje cell. These are the same neuron, the first neuron overtrained early learning. As in the Purkinje cell, you can see that the pattern on activity is different. Look at the first neuron during early learning. There is not this peak anymore of both left. I mean, it's and then kind of this bump after the movement. In the second, again, in the early learning, there is not anymore this peak. So the profile is completely different for left and right, and the same for the third one. So even here, like in Purkinje cell, during early learning, there is kind of a rearrangement of. And this is late learning, the same neuron. So the first one, the first neuron seems that's a kind of approach the overtrained task, especially for the right hand, even if it's not completely. The second neuron seems to approach, but probably the number of trials and in this case, and the third neuron seems to again move from early learning and a kind of activity that can be later, higher, similar to the overtrained task. Now, we can say look later. This can be also the reaction times in this case. So this difference. But in any case, the activity and pattern shift from the overtrained to a new task. And it changes also the pattern of activity. So even here, motor response is the output of the motor response of the motor. So even here, we have that's as in Purkinje cell, we did exactly the same. So the decoding on left and right hand in the overtrained task, same. There's a neuron aligned with the stimulus to the left, and the movement, sorry, is not aligned. The movement is the red. Okay. Even here, you see there is a dynamic across the population of the time, temporal dynamic of the time when the population identify if it's the right or the left association. This is early learning. In early learning, there are less neurons. Okay. And then again, but it seems that even in early learning, there is this kind of, again, these are all the p-values. So, I mean, it's a shaded of, probably I have to put only the the significant p-value, because you see that it seems that it's dark, but I mean, everything below 0.5. So anyway, and this is late learning. The same neurons. Again, all of these three are aligned with the time when the peak, the maximum. Actually, what I did, I found the difference between, for all the neuron, maybe it's better if I also point this. So sometimes, of course, you can find a streak in which there is a difference between left and right at different time. Okay, so I don't know, in 1,000 milliseconds, and then after. So which one I take? So I put as a threshold, I take, okay, at least the difference between the left and right, the same for correct and wrong I did. So it has to be at least 160 milliseconds. Then when I find the streak, okay, I take as a streak, so this is when there is a difference, when the difference is higher. So the highest difference between, in this case, left and right. So this is my streak. I tell you, there are also some streak before, but I took this one as a method. Now, I don't know if, so even in the dentate, neurons differentiate between left and right-hand choice in a dynamic way. We did exactly the same analysis, the cumulative distribution of the latency when the neurons discriminate between left and right from the onset of the stimulus. Again, the black is the OT, and the red and green is the later and early learning. There is not much different, later early learning. I think that can be also because the way I put, but because I don't have enough number of, these are 53 neurons, okay? So this is an overtrain, and sorry, this is aligned with the stimulus, and this is aligned with the movement. So here, if I do a Kolmogorov-Smirnov task, after the onset of the stimulus in OT is significant, but at the movement, no. Even if it seems that's before. And that's the reason I think that in this case is related with the manual reaction times. In this case, so in OT is earlier because the manual reaction time is earlier. Because at the movement, there is not much difference across the population. Like instead of Purkinje say, yes, there was a shift. Here, no. So we can say that an overtrained task can identify the upcoming and choice significantly earlier after the stimulus compared to later, but not the movement. I didn't put the movement. So probably this is the manual reaction time. So, okay. Then the comparing with the Purkinje set. Okay, we already see that the Purkinje set discriminate between left and right. So again, it's before or after. So this is a cumulative distribution, the time when the neuron distinguish between the left or right correct choice in the overtrained task. In the data nuclear, that is the data I showed before, that is the magenta, and in the Purkinje set. So now, here it showed that this is overtrained task, okay? And in an overtrained task, there is earlier discrimination after the onset of the stimulus of the upcoming left or right choice. And this is significant. At the movement, it seems there is no difference, okay? It's fine. So at the moment, they approach. In early learning, again, there is a very high difference. So the dentate identify with common choice much, much earlier than the Purkinje set. Now, at the movement, okay, before you can see, aligned with the movement, at the moment, this is not significant, but you can see that anyway, it starts earlier. It's here it depends on the statistic, of course, but from the graph. So the statistics, if I do the statistics, is not significant. So the distribution of dentate and the Purkinje set at the movement is not statistic significant, but it's clear that before the movement, in any case, more neuron in the dentate are identified. And this is late learning. So this is not significant. And there is definitely no difference between Purkinje and dentate. So it seems that, I mean, in early learning, so the population neurons in the dentate seems to identify the upcoming choice correct here earlier than the Purkinje cell. Of course, these are being recorded. The task is the same at different times. So the next step should be to record the Purkinje and dentate together. So this is, of course, has to be take also granusalis, but anyway, it's so at the end, overall conclusion. So both the Purkinje cell and the neurodentate nucleus encode the choice of upcoming actions and the outcome of the previous trial. So both of them. So, and then in both of them, these signals, so the identification of which the choice encompass the entire trial. So I'm not confined to the specifics, so they type. But both signals occur earlier in the dentate than in Purkinje cell. So now, why can be this case? So we know that in this we have the internal excitatory and inhibitory feedback loops between the dentate nucleus and the cerebellar cortex. Can be, it's a question, this part of this feedback loop to the Purkinje cell. Purkinje, granular cell, of course, I'd say Purkinje as a cerebellar cortex that drives or helps to make, to learn the association. Not only also this feedback, so remember that receive directly from the same information that are received the Purkinje cell received directly to the dentate from the mossy fiber, and then in the Purkinje cell also, but there are filter, granular cell. Here it is that seems direct. We don't know the dentate. All of this has been done in mice, so it's a speculation that something like this happened also in the dentate. And then we have, let's say, the convergent input to the dentate from multiple cerebellum zone mossy. So suggests that it's a role of a hub for integrate diverse signal. And then what's happening, these are all hypotheses. So at the end, the final output of the dentate, nucleus may relay various type of information that is not limited to motor function, and then back to the cerebral cortex. Mickey Goldberg, and then I didn't put all the affiliation because it's too long. And then Vishnu is a student in the lab. She just joined the lab less than 1 year ago, and he recorded all the dentate. And Valeria, she did the analysis of the decoder. Thank you.
Thank you so much. I know that this was a last minute that you had to put this thing together. So I'm really grateful that you were able to present it. Maybe, so we're almost out of time, but maybe if there's 1 or 2 questions we can have for you.
I'd like to ask, what would you expect to see if there was a switch as there was in part 1 of the stimuli? Would there be maybe some enduring representation in the dentate?
Probably I didn't understand the question. Can you say again?
What would you expect to see if there was a switch as in part 1? Maybe like some enduring representation in the dentate?
You mean the switch
Of the stimuli.
Of the stimuli? We did a switch here. We
Okay.
did a switch. So we also have reversal. I here didn't distinguish between new task and reversal because I took a very big chunk of trials. Okay, but of course we have to also analyze at the switch. I tell you what, again, one of the issue in this case, in this task, is that is the number of trials, because I mean, after the switch, we have, I don't know, can be 4 or 5 trials, and in 4 or 5 trials, correct and wrong, can be 3 correct and 1 wrong. So my issue is about again the number of trials we are allowed to analyze. So at the real switch, I mean, we have to do trial by trial. It's very noisy, of course, and from the statistic, it's a statistic point of view for me. Okay, the issue. So we're thinking about to give a more difficult task because he learns so quickly, and then so probably we can have more trials to analyze in very early part of the switch.
Right. Well, thank you so much. Thank you everybody for joining us.
Thank you.
I'm grateful and have a wonderful day.
Thank you. Bye-bye.